This is Chapter 3, Lecture D, Cytoplasmic Organelles and the Nucleus. Cytoplasm is the cellular material between the plasma membrane and the nucleus. So recall that when we think of a cell, remember the inside is called the intracellular fluid, the outside is the extracellular fluid, and so we're going to be focusing on the cytoplasm, which essentially is the intracellular fluid. This is where all the cellular activities are going to be found. It's where the nucleus is. And there's three main components to the cytoplasm that we need to be aware of. The cytoplasm contains the cytosol. And the cytosol, in Chapter 2, you learned that it was a colloid. And it's largely going to be water. It's mostly water. It also is going to contain important proteins. Remember, the building blocks of which are amino acids. There's lots of salts. In particular, there is lots of potassium in the cytosol. And there's also some sugars and some other solutes as well. The second part of the cytoplasm is going to be the organelles. And the organelles are really the metabolic machinery. So any time that something has to be broken down, that proteins need to be made, that ATP needs to be made, it's going to happen from the organelles. I like to think of the organelles as little organs. They're all going to have specific functions that we'll talk about here coming up. The third component of the cytoplasm is going to be the inclusions. And the inclusions, they may or may not be present. It depends on the type of cell, but they could have stored nutrients. For example, glycogen, which is, remember, a polysaccharide. There could be glycogen granules that would be stored in places like the liver as well as the muscle cells. There could also be an inclusion which contains lipid droplets, and these are obviously in fat cells. There could also be, in particular, examples of melanin granules that we might find in the integumentary system. So inclusions storing melanin as well. Now when we think about cytoplasmic organelles, they can either be with or without membranes. So they could be non-membranous or they could have membranes. The ones without membranes, in particular, these would be things like the cytoskeleton, which is really, you can kind of think of it like the bones of the cell. Could also be things like centrioles. And another example could be free-floating ribosomes. These would all be the non-membranous ones, the ones that do not have membranes. However, the large majority of them are going to have a membrane, and these would be things like peroxisomes, which we'll talk about in a couple slides. They could be things like lysosomes. It could be things like the endoplasmic reticulum, which we can abbreviate as ER. And another example would be the mitochondria, as well as the Golgi apparatus, just to name a few. So our next slide is on the mitochondria. The mitochondria kind of looks like a kidney bean, if you will, and we can think of it like the power plant. And it's going to be responsible for making or synthesizing ATP. So very, very important. And many cells that are metabolically active, that are very, very busy, for example, a kidney and a liver cell, these are going to have a lot of mitochondria, as you could probably imagine. So mitochondria is very, very specialized, and this is because it has, first of all, an outer mitochondrial membrane, as you can see on this slide, the outer mitochondrial membrane. It's also going to have an inner mitochondrial membrane here. And these inner mitochondrial membranes are going to form shelf-like projections, which are called cristae. And you can see an enlargement of the cristae in the lower left-hand corner of your slide here. And the cristae, they protrude into a space called the matrix, which is the very center of the mitochondria here. And the matrix is a gel-like substance, and there are specific fuels that are actually broken down here, like glucose. So for example, glucose is eventually used in our body to make ATP, and this chemical reaction is going to happen in the mitochondria. So glucose eventually is made into ATP. This process here is a metabolic pathway, a very, very important one, called cellular respiration. And since oxygen is utilized for this process is actually referred to as aerobic cellular respiration. Now, mitochondria are one of the most complex of all the organelles that we have. They are special because they actually contain DNA, RNA, and some ribosomes. So they actually have the ability to reproduce themselves, which other organelles do not have. So they have their own mitochondrial genes, which is really something very specific, and they're thought to actually originate and be very similar to bacteria and have their own phyla, which is the biological organization. So mitochondrial DNA are actually bacteria-like, which is quite interesting. It's kind of believed that mitochondria arose from bacteria that were invaded by ancient ancestors of plant as well as animal cells. Our next organelle is the ribosome, and the ribosome is very important for protein synthesis. And ribosomes can either be by themselves in the cytosol, so they can be non-membranous, or they can be attached to a membrane, as in the case of the rough endoplasmic reticulum that you can see here on the slide. So the rough endoplasmic reticulum is going to be very important for protein synthesis. As I said, we could have free ribosomes as well that are floating around, or membrane bound. So we're going to spend just a few minutes talking about this endoplasmic reticulum. As you can see, it's a very extensive network of tubules. So it's kind of a network of tubes, and it could either be studded with ribosomes or it could be without ribosomes, as you see here in the smooth endoplasmic reticulum. So smooth endoplasmic reticulum means that it is without ribosomes, whereas if it's rough endoplasmic reticulum, it means it does have ribosomes. And so the rough endoplasmic reticulum would be the site of protein synthesis. So once the proteins are made in the rough endoplasmic reticulum, they're then going to be prepared to go to the Golgi apparatus, which we'll talk about next, where the packaging, processing, and sorting of proteins actually happens. So smooth endoplasmic reticulum has some different functions. Again, it's important to remember that there is absolutely no protein synthesis that occurs here. But some of the functions that do occur in the smooth ER are the metabolism of lipids, and there is cholesterol that is made here. There is also various steroid-based hormones like sex hormones. So for example, the testes are going to have a lot of smooth endoplasmic reticulum to make testosterone. There's also the smooth endoplasmic reticulum is also very important for the absorption of fats. So we would expect to see a lot of smooth endoplasmic reticulum in intestinal cells. Another function is detoxification. So the liver and the kidneys are going to have lots of smooth endoplasmic reticulum. And another is the breakdown of glycogen into glucose. And glycogen, remember, is the large polysaccharide which has many units of glucose, and it's stored in muscle as well as in liver. Now what I want you to see from this slide is the Golgi apparatus. And the Golgi apparatus is going to make up what we call the endomembrane system. We're going to go back to this in just a few minutes here. but the endomembrane system is the system of several different organelles that sort of communicate together. But before we talk about that, we want to talk about the Golgi apparatus. So the Golgi apparatus is going to be found in this area, and we're going to zoom in on it in this next slide right here. This is the Golgi apparatus, and the Golgi apparatus is going to kind of be, it's going to sort of act like a traffic director. So if you think about the people that work at the airport to direct the planes which way to go, this is what the Golgi apparatus is. It's going to direct cellular protein specifically. And the Golgi apparatus kind of works kind of like a FedEx station. Another analogy would be to compare it to the post office, because it's going to receive incoming mail. It's going to take that incoming mail and package it, processes it, and get it ready, and it will prepare it to be exported from the cell. So the mnemonic I like to remember is PPS, which stands for package, process, and sort. So what it does is it sorts these proteins and gets them ready to go to their final destination. So it modifies, concentrates, and packages these proteins and lipids to get them ready to be shipped out. Now this slide is showing you several different destinations for the molecule based on where it needs to go. So the protein-containing vesicles that are leaving the rough endoplasmic reticulum where they were synthesized now are going to enter into the Golgi apparatus. The Golgi apparatus is going to take that protein, and the protein is going to travel throughout the Golgi apparatus here, and it's going to be modified so that it will eventually be ready to be shipped off to its final destination. So it's packaged into different vesicles, and there are different possibilities based on the contents of that vesicle. One pathway, pathway A, says that the protein could be exported for exocytosis. So let's say that this cell is making insulin, for example. Insulin is a protein. That insulin, once it's has been processed and modified, it now can be exported to its final destination, to the pancreas. Another possibility would be the vesicle contains proteins that need to be incorporated into the plasma membrane. So let's say, for example, that the protein is part of the sodium-potassium pump. Another example, this would be pathway C, is let's say that there is a protein that has been folded incorrectly. That protein, if allowed to go to its final destination, would cause more problems. So the way the cell protects is those molecules would be shipped to a lysosome for destruction, so they couldn't cause any further damage to the cell. The next slide is showing the lysosome, but before we talk about a lysosome, we're going to talk about another organelle, which is called a peroxisome. Kind of sounds like peroxide, and that will help you remember what its function is, because it has a lot of powerful enzymes that are present within this organelle, and two of these important enzymes are called oxidases and catalases. And so what they're going to do is they're going to use oxygen to detoxify certain chemical structures. For example, alcohol or formaldehyde. These are two chemicals that can be very, very harmful to the body. So the peroxisomes are going to destroy these. Another thing that they do is they neutralize free radicals. Free radicals can be very damaging to the body if allowed to stay throughout circulation. So they neutralize these free radicals. And what they do is kind of interesting. They convert the free radicals into hydrogen peroxide. And the hydrogen peroxide then is not going to be as damaging as the free radicals would be. So these peroxisomes, we would find them in certain tissues like the liver, as well as the kidney cells that play a very important role in detoxification. Now the slide that you see contains lysosomes. Lysosomes are going to be larger than peroxisomes, and they're going to have certain digestive enzymes. And so certain types of white blood cells, any sort of cell that has to dispose of invading bacteria, would have lots of lysosomes, and they would have specific chemicals that are going to destroy the cellular debris. So one example of lysosomes is cells that need to be the demolition crew, the ones that need to digest bacteria or viruses. There's also some that are going to actually be part of the bone that are going to play a role in digesting bone and releasing calcium into the blood. They're going to be very important for digesting non-useful tissues, again, so they don't cause more damage to the rest of the body. So lysosomes are very, very important in order for the body to stay healthy. So our next slide now is on the endomembranous system. And the endomembranous system is really a system of organelles. And it's going to be a system of organelles that is going to produce, store, and export these biological molecules. It's going to break down harmful substances, and it's going to first of all include the ER. It's going to include the Golgi apparatus. It's going to include secretory vesicles as needed, transport vesicles. So it's really kind of a group of organelles, membranous organelles throughout the cell that help to make sure that helps to ensure that the cell is functioning correctly. Our next slide then is showing the cytoskeleton. The cytoskeleton, you can think of it just like the cell skeleton. So it's kind of like the bones of the cell, if you will. And specifically, it's going to not only act as the bones, but it's also going to be the muscles, and it's going to be the ligaments of the cell. And this cytoskeleton has three main components that you see on your slide. It has the microfilaments, the intermediate filaments, and the microtubules. Two of the important microfilaments that we're going to deal with this semester are the actin and also the myosin protein. And they're very important for contraction. In this beautiful slide that you have here, they used a dye to actually show these microfilaments as a blue network surrounding this pink nucleus. And so they work with cell movement or cell motility to help the muscle actually contract. The next part of the cytoskeleton would be the intermediate filaments, and these are going to be important in attachment. Particularly, they're found in the cell junctions that you learned about earlier in this chapter, called desmosomes. And they can also work as neurofilaments in nerve cells and keratin filaments in epithelial cells. And then the last one would be the microtubules. These are the largest of the three different parts of the cytoskeleton. And they're going to be particularly important for the centrosome. The centrosome is the cell center, and it is a part of what are called the centrioles. And these are also going to be very important for movement within the cells. So organelles like mitochondria, for example, organelles like lysosomes, and other vesicles are actually going to be able to move throughout the cell with the use of these motor proteins, these microtubules. So these microtubules actually kind of work like a roadway system. You can kind of think of them like the railroad tracks within the cell. And they're going to allow for movement within the cell, which is pretty amazing. And our next slide is showing you an example of this. We have a microtubule here, which is our railroad, if you will, and a vesicle is able to attach to this microtubule, and it can actually walk to another part of the cell, which is pretty amazing. And you can see at the bottom of this slide that we can see the cytoskeletal elements, the microtubules or the microfilaments, allowing for the cell motility to occur. Now the centrosome, which is made up of these microtubules, is going to be found making up the centriole. So basically, a centrosome is a pair of centrioles. The centrosome matrix is really important, especially for generating the microtubules, and also it's important for organizing the mitotic spindle. And we're going to be talking about that in a subsequent lecture video. But the mitotic spindle is going to be very important for division of the cell. Other examples of how the cytoskeleton is very important is that it makes up cellular extensions that we see on this slide, like cilia and flagella. So cilia, first of all, is seen in places like the respiratory tract. So we have it in our bodies lining our trachea. And what it does is it helps to propel mucus that contains bacteria out of the respiratory tract. So we can either spit it out or we can swallow it. So it helps to protect us. So it gets rid of this mucus. And the really cool thing about the cilia is the centrioles are going to form the base of the cilia. So it's formed by microtubules. And the cilia is going to kind of move in a rhythmic motion, kind of like you would see the grass move on a very windy day. So it moves in rhythm back and forth, up to 10 or 20 times per second. Just phenomenal. And so you can see in this case there's a propulsive stroke and then there's a recovery stroke when the cilium moves back to its original position. So in this case, you can imagine in your trachea there is mucus that is moving from the base of your trachea towards your throat so that you can spit it out, or you could alternatively swallow it. Now, the microvilli are something a little different. The microvilli are important because they are going to increase the surface area of the plasma membrane. And what this does is it allows for more absorption of molecules across the plasma membrane. So we would expect to find the microvilli in absorptive areas of the body, so places where there needs to be a lot of molecules that actually go from the digestive tract into the cells for processing. So one example of this certainly would be in the GI tract that we would find the microvilli. There's also flagella, and we don't have a picture of flagella on this slide, but the flagella is going to be longer than cilia, and the only example of a flagella in the body would be the sperm cell. So it allows for propulsion, so for the sperm cell to actually be able to swim towards the oocyte. Our next slide is showing the control center of the cell. This is the nucleus, and the nucleus contains all of the genes. So it contains the genetic makeup that the cell is going to need, all of the instructions that are needed to make all the necessary proteins. Most cells are only going to have one nucleus, but some cells can be what are called multi-nucleated, and so this means they have many nuclei. Examples of these could include skeletal muscle cells, which you'll learn about later in the semester, a lot of times liver cells, and some others are going to be multinucleated. But all the other cells at least have one nucleus, except for cells that are anucleate. And if they're anucleate, this means they do not have a nucleus. And one example of this would be the red blood cells. So this would be cells that then can no longer divide. So the main structures of the nucleus are the nuclear envelope, or the nuclear membrane, the nucleoli, and the nucleoli is plural for nucleolus, and then also the chromatin. And the chromatin is going to be DNA here, and it's actually going to condense into another form to make the chromosomes when the DNA is actually ready to divide. So the nuclear envelope is a double-layered membrane, and it's going to protect the nucleus. It's going to be selectively permeable to only allow certain molecules in and out of the nucleus. So that is absolutely crucial. There are nuclear pores. We can see the nuclear pore complex here. These are just little pores that only allow certain molecules in and out. And one very important molecule that needs to exit out of the nucleus would be the mRNA, which is the copy of the DNA. And then all of the other jelly-like fluid that we see would be called the nucleoplasm. So instead of it being called the cytoplasm, which is outside of the nucleus, the nucleoplasm is the jelly-like fluid which is found in the nucleus. Now the nucleoli, or nucleolus for singular, it's going to be containing ribosomal RNA. And it would be the site of synthesis for ribosomes. The ribosomes structurally are made up of rRNA. So our next slide now is showing chromatin here. And the chromatin really you can kind of think of it like the thread which would be on a spool of thread. So the spool that it's on is actually a structure called a histone. A histone is a protein, and it's really kind of, it allows for the physical organization or the physical packing of these long threads of DNA, because otherwise the DNA would be very disorganized and all tangled up within the nucleus. So these histones are very, very important. And you can also see the DNA. The DNA is this double helix, and the DNA is the genetic makeup. Remember in chapter 2 that you learned that is an example of a nucleic acid. So it's very important that the DNA be preserved. And so what's going to happen is when the cell is not dividing, the DNA is going to be in the form of chromatin. And this is what you see here at the top of the slide. And over on the left-hand side, you see the chromatin here. However, when the cell divides, when it enters in what's called mitosis, which we'll talk about in a subsequent video, what's going to happen is the chromosome is going to condense into chromosomes. And this is very important because the DNA has to be organized in such a way so that it is equally divided for the future daughter cells. And your textbook does a great job of summarizing all of this, so especially the different parts of the cell, so be sure that you look at those tables in your textbook.